Okay, great. So first of all, thank you, Athena, and everybody participating on this set of seminars. It's been really great to listen to all this work while being in quarantine. So as Athena mentioned, the research that I'm going to show today to you guys is part of my PhD work. And here we analyze the early stages of adaptive radiation using as a model system the Tetra spiders from Hawaii, and as a technical approach, exon capture. Next. So the adaptive radiations are perhaps, at least for me, one of the most spectacular events in evolution. They correspond to the diversification of a large number of species in a very short period of time. And associated to that diversification, there is some key innovation that allows the exploration of different microhabitats to those closely related species. Adaptive radiations happen in different groups of animals, plants, in different timescales, in different geographies. But what is in general common of all of them is that they happen in isolation. Next. One very interesting observation that could reveal something about the temporal dynamic of adaptive radiations was published in 2007 in the book of Island Biogeography in a chapter written by Rosie Gillespie and Bruce Baldwin. Here on the left side, you will see the number of species standardized by area against the age of the island. This is for Hawaiian radiations. The left side is for animals, then the right side is for plants. And what we see here is for groups for bigger radiation, big number of organisms, of species, there's a peak in diversity in the Middle Age islands. So from left to right, what you will see as the second or third column of dots. And that peak on diversity then declines towards the oldest island of Kauai, which is the one in the far right. That pattern is valid for animals and plants for big radiations, yet for smaller radiation, there is some sort of monotonic increase of the diversity towards the oldest island, which I would consider some sort of null model, like what you expect. The older the environment, the more species accumulation. But what happened? What could we learn about the temporal dynamic of this explosive diversification by the fact that in the Middle Age island we have more species? Next. So the Hawaiian Islands have been considered historically as an example of a chronosequence where you start in the young island, the big island in the south, and as you move north, or as you move northwest, you're finding older and older island, which could be considered as some sort of snapshot of a process in time. So if you, let's say, stay on the big island, you will see some number of species. Then as the big island becomes older, let's say getting 2 or 1.5 million years, you will expect to see a peak in diversity, and as become even older, like Kauai, a drop. So is that the case? And what is this revealing about the trajectory, the diversity trajectory of an adaptive radiation? So that's kind of the big picture where this project is framed. Next. To study this, I'm introducing the Tetra group in Hawaii. These spiders represent, on this tiny archipelago, about the 11% of the world's diversity on the group, and there are many more species to be described. Ecologically, they are way more diverse than any other Tetra in any other continent, so it's very special. Next. They are the result of 2 independent adaptive radiation, the spinal leg clay, the one that I'm going to be talking about, and the web building clay with more species, and it's a group that still spins webs. Next. The spiny leg spiders, they have 4 different ecomorphologies: green that lives on leaves, maroon that lives on moss, big brown on bark, a little brown on twigs. These 4 ecomorphologies evolved semi-independently in all the different islands. You never find 2 species of the same ecomorphology living together. So we can say that this is a well-behaved, quote-unquote, adaptive radiation. So that situation is valid for different islands / volcanoes, except on Haleakala, on Maui. So here you can find 3 species of the green ecomorph living in a parapatric condition on the same volcano. Curiously, we are finding this on a middle-aged island. So it kind of smells that we are finding at some sort of transitory stage where we have this peak on diversity. Next. So looking at this situation more closely, here we see the 3 green ecomorph species. They all have species on Haleakala, which is the volcano on the center on the upper center part, you see the 3 dots, and they all have a second population or a second set of populations on a different island or volcano. Next. So the question is how this pattern came to be. I'm going to present 2 extreme hypotheses. One of them is this one, where we can imagine an ancestral species with 3 populations on different volcanoes on Maui Nui before Big Island existed. These 3 populations, next, will turn into 3 different allopatric species, and next, they will move around and reach the current distribution. As a second hypothesis, next, we will imagine that we have 3 populations of the same ancestral species that by parapatric speciation turn into 3 species, next. And those 3 species will then move around and reach the current distributions, next. So what I present to you are the 2 very extreme cases. Most likely what could have happened is something in between, where, for example, you have 2 species evolving in parapatry and 1 in allopatry, and also probably all the species colonize all the islands, but only 1 establish a permanent population. So for instance, if you do some sort of admixture analysis in a population in West Maui, the blue dot that appears by itself, maybe you will have some contribution of orange and also green. That could be a situation. Now, the way to tease apart these 2 hypotheses, or maybe figure out something in between, is basically doing a very deep characterization of the population history of these 3 species. Next. So the way of how we addressed this was with exon capture. First, of course, and here's kind of like a flow chart of what we did. First, going to the field, collecting the specimens, doing library preparation, following Mayer-Kircher protocol. Then on the left side, there is like another set of work, doing a whole transcriptome sequencing of 1 fresh specimen, designing probes, and then printing them on a chip. We did 3 experiments of hybridization on a chip base. So this is 2013. So it's a little bit a while ago. A third leg of this hybridization is the design, the in-house design of a Cod1 library to block Cod1 DNA, which could affect the hybridization. So then we have all our libraries, was about 150, 3 chips, Cod1 library. We do the hybridization. Then we run a set of post-control tests, for example, qPCR for positive controls to check for enrichment, for negative controls to check for depletion, bioanalyzer trace to look at the fragment distribution and making sure that we don't have PCR duplicates or other things that will reduce our sequencing power. Once everything looks fine, we send this to a high-sig machine, we get the data back, and then we run the raw data set through a set of cleaning steps, which are basically the ones established by the Museum of Vertebrate Zoology at UC Berkeley. On their GitHub site, you can find them. So basically, we follow all of that. At the end, we use a SNP cleaner. And then the final data was analyzed with 2 different strategies, both of them using ANGs as a first step. In one side, we determined the side frequency spectrums to get admixture plots and PCAs. And on the other side, we actually call SNPs. So it's a different approach, right? In one case, side frequency spectrum, and in the other, getting the SNPs. So we got the SNPs to do a split tree. Next. So remember, this is kind of the biological situation, and we want to understand how this happened. Next. So the first approach is a very exploratory PCA analysis. Let me walk you through this plot. Here you will see the shape of the dots. Each dot represents 1 individual. The shape represents the identity based on the locality. Remember, we don't have evidence of more than 2 species on the same site. So in principle, being very naive, you can think that the locality could give you the identity. Then the color of the dot represents the identity based on morphological identification. So you can see in the upper part the color that represents each species. And then in gray, all the juveniles, because juveniles cannot be identified on this group of species. So first surprise, we have 3 species, but we find 4 very well-defined groups. So on the upper right part, you will see 2 clusters of green and gray species. The one on the far right corresponds to all the specimens from the Big Island. Here you will see gray and and green circles, which means adults and juveniles of Tetranatha brevignatha from Big Island, nothing special. The interesting thing is they are significantly separated to the ones from Maui. So even if brevignatha it's supposed to be in both islands, genetically they seem to be as separated as what you would expect from different species. So first surprise. Then if we move counterclockwise, we're going to see another group of orange and gray squares. So square because those are the sites where you expect to find Macracantha. They are orange because some of them are adults, so that's fine. And then we find also orange diamonds. Those because we included in this study some individuals from localities that were not surveyed before. So we didn't have a prior of what it could be, but here we can see that they're all Macracantha. Not a big surprise. Something interesting is Macracantha has two populations, Lanai and Haleakala, but on this PCA analysis, they don't separate as much as the two populations of Brevignatha. So that kind of gives us a hint that the separation between the two populations of Brevignatha is higher than the ones in Macracantha. But things that really interesting when we look at Waikamoe, this is blue triangles. So if you look at the lower left corner, you will see blue triangles, so those are adults of Waikamoe living where you expect, that's okay. Then gray triangles, so those are juveniles. But then we also find blue squares. A blue square means that adult was morphologically identified as Waikamoe, but it was found on a place where you expect to find the other species. Genetically, of course, it's clustered with the rest of Waikamoe. So basically, it is an individual of Waikamoe that was occurring in sympatry with the species Tetranatha macracantha. But that situation also happens with Brevignatha. Remember the circles. If you look at the far lower left corner, you will see two gray circles. So those are juveniles of Waikamoe located where you expect to find Brevignatha. So the main finding here is Waikamoe is actually occurring in sympatry with the other two species. So this is not so well-behaved adaptive radiation. We have two ecomorphs coexisting. Next. If we zoom in only in Waikamoe, we will see, so we do like a PCA just for that species. We see the separation between West Maui and East Maui. West Maui only triangles, so those are all Waikamoe specimens. But where the overlapping between the other species happens is actually on East Maui. Next. So once you have two species recently, or three species that derived very recently, couple million years, occurring in sympatry, a natural expectation is probable hybridization. So we did an admixture analysis. Next. And here I'm highlighting in these red squares the two populations where we find the overlapping. And you can see that each one of them, Kipahulu and Lower Waikamoe, you see two solid color, two set of bars with solid colors, implying that there's no hybridization. So we have overlapping occurrences of two species of the same ecomorph without hybridization. Next. Now, we wanted to double check that not using side frequency spectrums, but actually calling SNPs. And here's a split tree, a phylogenetic network that shows the distribution of all our specimens. In the lower part, all the species from the Big Island, clearly separated from the ones on Maui, which already we saw on the PCA analysis. In green, in the upper part, we have the Tetranatha brevignatha from Maui, clearly closer to the other Maui species instead of the one that is nominally the same. Brevignatha from Big Island. In orange, we find Macracantha. Even if in the PCA they all appear clustering together, this analysis allows us to see a difference between the specimens from Lanai and the ones from East Maui. So this is kind of a sanity check. And then when we look at Waikamoe, we see a common stem and then a strong separation between the West Maui and the East Maui specimens. Now, all the individuals that occur in overlapping distributions, the potential hybrids, they appear clustering in blue. So there's no hybridization with this other method. Next, please. So what we learned so far? The original three nominal species cluster in 4 or 5 clearly divergent genetic lineages. Also, the populations of Brevignatha are genetically as divergent as what you expect for 2 different species. In the third place, Waikamoe is actually a widespread species present in different parts of East Maui and not restricted only to where we initially thought it was. And finally, there's no hybridization between these close related species. So what this all means, next. is that here we're showing evidence of sympatric distribution of closely related species from the same ecomorph without hybridization. Next. This situation, initially thought as impossible, actually has been discovered very recently in other well-studied adaptive radiations like the anoles in the Caribbean or cichlids on East Africa, revealing that a deeper study of the genetics of these organisms and more fieldwork allowing to identify their proper distributions shows this kind of gray area, this in-between stage where reproductive varies already established, but ecological differentiation not. Next. So what we propose, what we think is going on here is the following. Imagine you have an island with a volcano, with a patch of rainforest, with one species. As the island becomes older, more volcanoes appear, let's say 4, and each volcano has its own population of this one species. So we have 4 populations. If there's no gene flow, these 4 populations might turn into 4 species. Next. So we go from 1 species to 4. So this is increasing diversity. 4 allopathic species with this set of islands within islands. As the island gets older and more time passes, things like movements between volcanoes could occur. Next. And we'll have this situation. And next, if the island gets even older, some of the endemic habitats for the species, in this case triangle and a star, might disappear, but the species actually survives in another place, staying in a situation of secondary contact with a former sister, well, with a sister species. So this slide that I'm showing here, this cartoon, is actually what we present evidence now in this study, the actual secondary contact with these 2 species. Now, what is coming next, Athena, next please, is what I will say some sort of character displacement at an evolutionary level, where after these 2 species having the same ecology come together because they cannot reproduce, they differentiate ecologically. And next. So this will result in something like this. From a common ancestor in a, let's say, green ecomorph, we have these 4 species closely related, but as you look at pairs, they might have a different ecology, a different ecomorphology. So again, this change in the ecology, we are not providing evidence from this data set, but is what we expect, what we predict to be the next step to explain why we have convergent ecomorphologies in this set of islands. Next. So just to finish, a large number of people who helped me even during the field work, getting permits, doing lab work and data analysis, and many, many sources of funding. So next. If you are interested in this, this work is already published, and I've noticed that a lot of the talks are strongly technical, so I will recommend, if you are interested in the more technical aspect, to look at the supplementary material number 1, the file number 1. That one explains extensively all the details of how we move sequences from here to there, and might be useful for other type of projects. So that's what I wanted to show. Thank you very much.
All right. Thank you, Darko. To Michael. Okay. Matt has a question.
So great talk, Darko.
Thank you.
Do you think that you would be able to detect these same sort of patterns in much older island arcs? Like, let's say those islands were around for another 10 million years. Would it be possible, you think?
So, well, a couple of things. Usually, if you look at the age of volcanic islands in rainy areas, like Central South Pacific or some of the islands in Macaronesia, they don't go beyond 7 or 5.5 million years. Some of the Canary Islands, where it rains less, they can reach, I think the oldest of the Canaries is 8 million years or something like that. So that's one restriction. Then what we are presenting here is what we think is happening in middle-aged islands, where we find this kind of peak on diversity. So if the islands were to get older, like in the Canary Islands, I would imagine that what you will see, it's a tendency of more and more extinction. So the extinction rate will become more important than the speciation. That also because as the islands become older, they start shrinking, losing habitat. So some of the drive for speciation, especially the ones related with the geography, would disappear. And as the island shrinks, the possibility of the initial step of allopatry would not exist. But that's just speculation, right?
Oh, very cool.
Great. Thank you, Darko. Any more? Cynthia has a question.
Hi, thanks, Darko. I wonder if global warming plays a role in your results, and how are you correcting for that noise?
Global warming. Oh, I see. Okay. I'm not... Okay, if we talk about global warming, the main effect could be related with changing rain patterns on these forests, perhaps moving the forest upwards or things like that. There's no evidence that since global warming has become important, like industrial revolution, there has been a relevant shift on vegetation lines. But what maybe has a strong effect, I'm not accounting explicitly, is related with, like, let's say, climate change in a more broad sense, and it's the extinction of all the low elevation species. So a lot of what we know about volcanic islands is usually the story of higher elevation because there's less human impact. That's why many studies are about cloud forest. But lowland rainforests in these islands is very unknown. So everything we understand is kind of missing. There's this bias extinction rates. Now, for my particular study, because I'm broadly talking about close related species and the lack of hybridization, it kind of really doesn't matter if I'm missing other close related species. The statements stay the same. But maybe there were most likely there were more species involved in this group.
Thank you. Thanks.
There's one more.
One more question for you, Darko. Can you explain any more about your analysis in split tree to assess hybridization?
Sure. So on a split tree, let's say if you have two branches, right, like this. If there is some hybrids, you would imagine that they should be placed in between, basically, because they will share some SNPs with one species and with the other. Basically that.
Great.
It's kind of like on a PCA, if you have one species here, the other here, and there are hybrids, the hybrids should be in the PCA space kind of in between.
Okay, any last questions for Darko? All right, thank you, Darko. Darko will be back again next week as well to talk about his spider genomes. Okay, our next speaker is Isaac Marck. Isaac is a grad student at Berkeley, and he's going to talk about rock daisy phylogenomics. Isaac, we can see you, but we can't hear you.
Can you hear me now?
Yes.
Okay. Can you see the slide?
Yep.
Okay.
Maybe make it full screen.
Yeah, okay. Full screen. So you can hear me and see everything. So,
Yeah.
Okay. I'll just get started with saying thank you, Athena, for organizing this, and for the Matt and Darko for giving great talks, and also for filling in some of the background for the talk I'm going to give. This talk is called Phylogenomic Insights into the Systematics of the Rock Daisy, Perityleae Compositae. My name is Isaac Marck, by the way, and I'm a fourth-year grad student at Berkeley in the Baldwin Lab. I work in the UC and Jefferson herbaria, where I study the evolution of flowering plants with a focus on the sunflowers and daisies, which is one of the world's most diverse plant families. Accounts for about 10 or 12% of flowering plant diversity. And I'm really interested in these plants, because they're so diverse, but also because they have a really interesting ecology. And it's also a family where molecular phylogenetics has kind of upended a lot of long-standing ideas about deep relationships. and also put into perspective a lot of those morphological traits that were previously important for taxonomy. But in the same way that molecular phylogenetics had this big effect, all of the phylogenomics approaches that we've been learning about in the social hour have the same kind of potential because they, increase our support for hypotheses, but also really important for working in plants. They have the potential to resolve conflicts when incongruences arise between different sources of data, organellar and nuclear sources of data. And so I'm going to talk about the work I've been doing in a group of composites that hasn't had a lot of previous phylogenetic research using molecules. The group is called the rock daisies tribe Perityleae, and it's a diverse tribe of herbaceous and perennial plants that are found in arid environments in Western North America. And members of this group are parts of the abundant show-stopping wildflower blooms that happen in the desert after rain. But what really interests me about this group is that a majority of the plants are these perennial shrubs that are extreme ecological specialists because they grow on vertical rock habitats. So the plants themselves are, they have a woody caudex that's rooted into the cracks and crevices on vertical cliffs, and then it anchors the leaves and the flowers there, and they grow on the rocks, but they don't grow off of them. And because of this, the plants are, for the most part, restricted to high altitude sky islands in desert habitats. So they're kind of isolated onto the tops of these mountain ranges in the deserts in the southwest U.S. and also northern Mexico. And they have a lot of elements that are similar to a kind of island-like radiation, where you have a lot of plants that are isolated to just one mountain range. And then, that also means there's a high proportion of this group that's endangered. At the same time, they have received very little past study using molecular phylogenetics, and the limited sampling that's been done was limited by support. Because they're perennial shrubs and they have, long generation times, high copy targeted loci haven't been so useful in resolving relationships. So I started several years ago to investigate these and took a genome skimming approach where I visited herbaria and collected material from historical specimens, little bits of leaf material for DNA analysis. And these are actually pictures from a visit that I made with my collaborator, Will Freeman, to Cal Academy, where the staff was very nice and let us study the specimens and then took us out for delicious Chinese food. And then the picture on the top is a picture of me doing lab work in the EGL. You can see from my face how much fun I'm having. And the genome skimming approach is an approach where you basically shotgun sequence material, library prepped material. We sequenced at 100 base pairs paired end, and then you use reference guided assembly to assemble the high copy gene region. So in our case, we targeted the chloroplast DNA and the nuclear ribosomal DNA, as well as mitochondrial DNA. And I'll show some results. Basically, what we did from this was to make two phylogenetic trees, one based on the whole chloroplast genome and one based on the whole ribosomal genome.
So I'll go back to here. So this is showing a tree of the chloroplast and a tree of the ribosome. And I was making the point when I think I was cut off that the two trees are really well supported, but they disagree to a large extent about the topology of the tribe. And this is a conundrum, right? Sometimes similar clades are resolved in either tree, but they come out in different places. And in other cases, a tribe is resolved in the ribosomal tree that in the chloroplast tree is not resolved as a natural group. It even looks like the names have just kind of been thrown onto the tree. And this is a general pattern here. So it was a conundrum because we had two well-supported sources of data, but they conflicted as to hypotheses about the topology of the phylogeny in this group. So not knowing what to do, I went to the desert and I went and studied these plants in their natural habitats. And what I did was kind of just to sweep across their geographic range and to climb up the mountains and study the plants in their environments where they could be found, and to collect material for DNA analysis and make herbarium specimens. And this was a lot of fun and very educational, but also a bit dangerous sometimes, as these pictures show, where you can kind of get into a free solo systematics situation. So as often happens in these big projects, when you don't know what to do, somebody shows up who has some important advice and helps you. And for me, that person was Dr. Vickie Funk, the Smithsonian curator and sunflower expert, who I met at a conference and who let me know that they were working at Smithsonian on a set of target capture baits that would target over 1,000 low-copy nuclear loci for the sunflower family that could be used for phylogenomic analysis. And she invited me to Smithsonian to try these baits, which were being developed for a family-wide analysis on at a tribal and generic scale in the rock daisies. After I was able to get support to go to the Smithsonian, Vickie ended up becoming very sick and passed away this last fall. So her inspiration is really important to me and to hundreds of other students. And so a lot of this research is dedicated to her memory. I'll just check and make sure, is everything working? Great. So I'll show some results. Okay, so in a similar fashion, on the left side here is a tree based on ribosomal data from the genome skim, and the tree on the right is based on data gained from that target capture approach. We targeted over 1,000 low-copy nuclear loci, but using Phylogenetic to assess potential paralogs in that data, we actually lost a majority of that data and ended up with a smaller sample of about 200 orthologous, putatively orthologous low-copy nuclear loci. But as you can see from the tree and the black dots, which indicate high bootstrap support, it's a really well-resolved phylogenetic hypothesis. And when compared to the ribosome, there's a lot of congruence between the two sources of data, although there's some differences there that are probably the result of higher support in the target capture-based tree. And I'll just highlight a few things that this new understanding of the rock daisies also comes with a better understanding of monophyletic groups, natural groups, which correspond in some cases to past taxonomy and in some cases don't. So there's some shaking up of the Perityleae world happening here. And most importantly, not most importantly, but just to illustrate that, the boxes shown here are all members of the same genus Perityleae. But the little purple box that's off on its own contains the type for that genus. So there need to be some taxonomic adjustments in order to accommodate that. And the result that I'm most excited to share, and really just kind of on the surface here, is that there's resolution of a monophyletic group, a natural group that corresponds to all of those Sky Island endemic lineages. And that sort of sets the stage for more careful dissection of the evolutionary diversification that's happened in that group, and modes and tempo. So some of the conclusions that a phylogenomic approach here has led to a better understanding of relationships in the rock daisy tribe, and some new perspective on the evolutionary basis of the taxonomic traits that have been used in the past for their taxonomy. But this is all part of a process of refining a new approach for phylogenomic understanding of Compositae tribes and genera that has the potential to be important, since it's such a big family and ecologically abundant family, especially in Western North America and the Americas in general. And then there's resolution of this clade corresponding to a Sky Island radiation that I'm excited to explore in more detail. So with that, sorry for the technical difficulties. I'll say thank you for listening, and thank you to all of my collaborators, the people who helped me in the lab and the field, also with data, my mentors, the herbaria who shared material with me and also my funding sources. And I put my contact information here so that if you'd like to talk with me afterwards, you can.